Hello everybody, we're starting chapter 7, Introduction to the Endocrine System. So this first picture is interesting on the left there. You see this is a very old piece of artwork here. This is pre-Columbian age of a stone carving, and it was considered very beautiful. And notice what this carving here has on the neck. It has a goiter. So like the person to the right of that, there is the enlargement of the thyroid gland. So notice in this diagram here, you have the normal size would be in red, and then the gray is the enlarged thyroid. Also another symptom of hyperthyroidism is the bulging eye here. So this is to introduce you to the endocrine world here, thinking about what happens when some of this goes wrong. If you take it back and look at how endocrinology started, how would you even begin to study hormones when they come in such teeny quantities? So the first observational studies, if you take it back to about 1800s, Berthold performed a classic experiment with roosters that became very famous. So what he could do was pretty dramatic. He could remove the testes and find out what kind of behavior ended up in that poor rooster. So it turns out that the rooster ended up being less aggressive, and it had a smaller comb. The next thing you'd want to do is put them back again. What happens if I put that amount of hormone that's in the testes back into this poor rooster again? And it would become more aggressive. So, indirectly is working with those hormones, but really with the entire gland. Experimental endocrinology then, in the late 1800s, a French physician, Charles Brown, he made a big announcement in the scientific community, and he claims he conquered sexual rejuvenation. And how did he achieve that? Well, he injected himself with water that contained ground up bull testes. So he took the testes, ground it up, mixed it with water, injected it, and his life changed. He claimed that his sexuality had been revived. And what's wrong with this thought? The first thing is that these hormones are not water soluble, so that he could have never made an emulsion that actually he could use. So the whole thing was silly, but whatever he believed was fine. So after that, organotherapy became fashionable. It's, can we take a gland from one animal and inject it in another one, or administer it in whatever form to another organism, and see whether the effect of that hormone truly had an effect on a different organism. So that question then arose, could we use animal extracts as therapy in human reduced hormone levels? Because we would have conditions where these hormone levels are low, and perhaps we could fix it. So the question is, could you cure hypothyroidism? So if the thyroid is underproducing, could you actually supply that individual with thyroxine, which was the hormone from a different animal? Could you do things like use animal estrogen in women, in females that were low in estrogen? Are these possibilities? And of course we know now that they are, but these were the first questions that arose when people started studying endocrinology. A good example to look at is insulin, because we all think about insulin and how individuals that are low in insulin have trouble regulating their sugar levels. And so the first sort of solution for this low insulin was obtaining it from other organisms, obtaining it from a pig, from a cow, or a sheep, and taking this insulin and being able then to administer it to a human. Of course, we've come a long way now, and the human gene for insulin has been inserted into bacteria, which then will synthesize the hormone, and then we have it much more cheaply, and we have insulin that can be then injected into an individual that has hypoinsulinemia. So as a summary, how have we gone studying this through time? Remove a gland and observe an animal. You can put that gland back and see what changes. You can implant a gland in a normal animal. What if an animal's already producing enough hormone and you give it more hormone? What's the outcome of that? How does the behavior change? Maybe you can isolate the hormone. Difficult, but you can, and we have, of course, right now. And can you synthetically reproduce the hormone? If you don't even have to isolate it, but you can actually make it in a lab, it becomes much cheaper. And then therapy with hormones has become much more affordable. So in the description of a hormone, what's a hormone? A hormone is a chemical messenger. And we are still discovering many hormones that we didn't know existed, or we didn't know that were affecting our body the way they do. So it's a very interesting open field. We know that they control our lives. Hormones change the behavior of cells. They change the behavior of individuals and control how that individual reacts to things. All sorts of aggressive behaviors or leadership behaviors. A lot of those are hormonal, which is kind of scary. So they control the cell, and I'm going to go back. They control the cell, and what we want to look at in a little more depth is how do they control the cell? What do they do? When a hormone arrives at a cell, it's going to make it behave differently. What is it that it's doing? So I'm going to tell you that it can control the behavior of a cell in 3 different ways. First way, it controls the rate of enzyme reactions. So some mechanism is the best word, is going to change once you have an enzyme that can speed up some sort of mechanism that's going to cause the cell to behave differently. So an enzyme is speeding up reactions. It can also transport ions or molecules across cell membranes. So if in order for a cell to react, it needs to gain calcium or lose calcium or gain potassium, that transport of ions is going to occur more rapidly or more easily once that cell is subject to that hormone. Okay, so transport. And the third is that it can control gene expression and synthesis of proteins. What does that mean? It means that it can take the DNA that's in that cell, and let's say there's a portion of DNA that's not being expressed, that's something that's not needed by the cell at a certain time. This hormone can trigger it kind of like an on switch and say, go, we're going to synthesize a protein. You make it, look at the DNA, make that protein, and release it. And that's going to affect other cells to change their behavior. So controlling gene expression and synthesizing proteins is going to have a great effect on the body. There's a little vocabulary that sometimes gets confusing. What's a hormone? What's a paracrine? What's an autocrine? And how are they different? And you know there's enough overlap that they become quite confusing. But I'm going to try and simplify it for you. Hormones are chemicals secreted by glands into the blood. Estrogen is an example. So it's made somewhere. It's going to have to be placed into the blood. And the blood is going to circulate, go throughout the body, and reach the cells that need to be affected by that particular hormone. A little bit slow because it's traveling to its destination. Now, very small quantities of these chemicals can cause a huge effect on the body, which is so very crazy. Tiny quantities. And so just think of them as they're secreted, they go in the blood, they get distributed to other parts of the body, and then when they reach their target cells, they're going to have an effect on those particular target cells. Not all cells are affected by the hormone that's been released, right? Estrogen doesn't affect all cells. It affects particular cells, and so it will travel in the blood and reach those cells that need to be stimulated. A paracrine, on the other hand, is also a chemical that's similar, secreted by a cell that's similar, but they act in the immediate vicinity of the cell that secreted that signal. So they're not going into the blood, throughout the body and reaching somewhere, but they are just secreted in that one area, in the neighboring area, like histamine, right? You swell up with some sort of you get a mosquito bite and you swell up, not everywhere, you swell up just in that location. So that would be release of a paracrine that's affecting that. An autocrine is also a local chemical that acts on the actual cell that secreted it. So not on the neighboring cells, but on the cell itself. And that would be auto-self-crine or paracrine. And this is T cells stimulate themselves by secreting chemicals. T cells are part of your immune system. So in some cases, the molecule can be both a paracrine and an autocrine. You can see how that it secretes it in the vicinity, but it also affects itself. So look at this diagram of an autocrine signal. It acts on the same cell that secreted it. So it's secreted from here, and it's the receptor for that hormone, that chemical, is on the cell itself. On the other hand, it can also secrete it to somewhere else, to the cells in the vicinity. So autocrine and paracrine. You need that receptor there, right, in order to react to that particular molecule. So a hormone, I'm telling you, makes a cell react. It's going to initiate some sort of action, and it's going to then end the action. And then that is over. So it has a beginning and an end. So given that cells need to communicate with each other, we know that there's some molecules like insulin. We're going to go back to insulin. And insulin does what? Insulin is very important in regulating sugar in your body. So in this diagram, I want you to see, let me change my pen color here for a minute. Here's the pancreas. And the pancreas is going to have two different types of cells here that secrete two different hormones. One of them can be insulin, one hormone, and the other one is glucagon. So under different conditions, it will secrete a different hormone. Under what conditions? Well, is blood sugar high or is blood sugar low? And under those different conditions, it's going to secrete a different hormone that's going to affect how all other tissue cells take in sugar or release sugar by the liver. So I'm going to stop this video now and start video 2 of Introduction to the Endocrine System using this slide as a start.